Hey there, ladies and gentlemen, boys and girls. Today we're going to be talking a little bit about a pretty tough topic. We're going to kind of simplify it a lot today, talking about spectroscopes and spectroscopy, which sound like both very, very big words. All right. I have to really emphasize right now, though, before we go into this video, make sure you have finished the lecture on light and color and understand the difference between, in terms of a wave, of a violet color wave and a red color wave, in terms of frequency and wavelength. If you don't, go back and look at that stuff, because then this is just not going to make any sense whatsoever. All right, so go if you're not ready. Okay, I'm assuming you're ready now. Okay, so again, we're going to talk about something called a spectroscope. All right, so let me just spell it out for you guys, because hopefully you're taking your notes with this. All right, and I'm not going to make you draw all of this right here. All right, I wouldn't recommend it, but because I just want to give you a big idea about what is happening here. All right, so basically what we have here is we will have some sort of light source. All right, we have some sort of light that is going to go through some sort of sample. By sample, that could be, we use this a lot in space science when we're trying to look at galaxies or stars very, very far away. And what we do is we shoot that white light through it. Right now, remember, white light is all colors, right? All the colors of the wavelength. Some of those colors are going to be absorbed by the sample, and some are going to be, allowed reflect or actually pass through it. All right. So you take the sample, you shoot white light, you see which ones go through, and some will go through all the way. Some will go through a little bit, like partially absorbed, and some of them will be fully absorbed and don't go through at all. And then they hit this slit here. It's like a lens. It's a graded slit, and it actually spreads out the light, right, into different directions. And then we can measure for each color, like wavelength, how much of it goes through. Maybe only 10% of the red goes through, but 95% of the violet goes through. All right, and maybe 20% of the green goes through. Every sample is going to give a different, what we call spectra. All right, and this is what we call a spectrograph right here. That's our spectrograph, and it's going to look like a graph. And all you have to understand to be able to read these spectrographs is about that different colors have different wavelengths, and those different wavelengths will go through or be absorbed or reflected by each object. So again, real recap before we go into what it looks like, a spectra looks like. White light goes through the sample. Some colors are absorbed, some are reflected. Then that light is spread out, and we see how much ended up going on the other side. All right, and so what that will look like is we'll get some sort of graph. All right, and so let's say we have a graph here, and so this is how much is being, let's pick a different color so you guys can see a little bit better. Oh, I can't write on this. One moment. All right, we'll use this one. All right, this right here is going to be the percentage absorbed. Yeah. All right, so by that sample. So what we might end up getting is something like this. We'll get a graph that looks like something like, it'll have little peaks and valleys, and then it'll be like, all right. So basically what that means is this object didn't absorb many light waves, except for one wavelength. It absorbed a lot, somewhere between 600 and 700 nanometers. And we know that something that kind of a long wavelength. So yeah, see here on the bottom here, we have wavelength. Something with a long wavelength, when it comes to colors, it's more likely going to be a red or an orange. So whatever object we're looking at is absorbing red or orange. And if we know if it's absorbing orange, that object is probably orange color. All right, so we do this a lot of times with different objects that we can't necessarily see with the naked eye. All right, and so sometimes you'll get even ones that are mixtures like this. So let's just pick a different one here. So let's say, let's pick this color. All right, we might see kind of a spike here. And so we might get two spikes here. All right, and so what we see here is that it is absorbing something at this wavelength here, which we know is about blue, and we are absorbing something at about that wavelength. So it's whatever this object is absorbing blue, and it's absorbing red. This will help us identify this object is not the same object as the last one because it absorbs a different wavelength of light. This is a good way to identify different materials. Different gases do this. So, for example, a good example that we actually use in space science is that if we did a spectroscope of the sun, all right, it would look like this. You would think it would look like yellow, but that's actually not the case. It's just like, oh, no, sorry, that's just, if you use a specific lens, we'll get this. Actually, what we get when we do the sun, my apologies, would look like this. Because it gives off white light. And white light is all the colors. So another way we can look at a real example here instead of me talking about it here is this is an example of chlorophyll. Now, chlorophyll is just a molecule you find in plant cells that are absorbing light so they can do photosynthesis. But there's two different types of molecules. So the kind of question I would ask, because this is kind of a complicated topic, the kind of question I would ask is, for chlorophyll A, what color is it likely to be absorbing? And we know that it's at the very end of this spectrum here. It's absorbing red. So this, or chlorophyll A is absorbing red. And if I said chlorophyll B, what color is it absorbing? You would say, well, chloroform B is absorbing not only here, but its biggest point is right here. It's absorbing like a bluish color. You notice there is one color that neither chlorophyll A or B absorbs, which means if they're not absorbing it, then they are reflecting it. And that is right here. This is all being reflected. All that light is being reflected. And that wavelength there is green. That's why leaves appear green, is because they reflect all that light and it goes to our eye. They are not being absorbed by chlorophyll A and chlorophyll B. Right? So this stuff can be a little bit tricky, but really what it is about interpreting a graph. Knowing that the spikes are how much that light is being absorbed, and knowing what that spike matches with its wavelength. All right. Now, I know this is a tricky video here. I'm going to put some supplemental stuff up there a little bit to help you out with it. But this is a tricky one. Make sure to do that Schoology practice. There's some practice questions on there about what I would kind of expect to be able to read. All right. Otherwise, watch over the video again if you can. It's a tough topic. Take your time. And good luck. And may the science be with you.